Okay, guys. Today I'm going to talk to you about RNA and transcription. Now, hopefully by the end of this lesson, you'll be able to understand how RNA interacts with DNA to make mRNA, which is part of the central dogma of molecular biology. Transcription is the process of mRNA being copied from DNA, and hopefully you'll be able to understand that by the end of this lesson. The role of RNA. DNA was discovered by Watson and Crick back in the 50s. And right away, they were able to see how the replication of DNA was evident through the complementary nature of DNA. Now, what this means is there are 4 bases in DNA, and they are complementary to one another. Adenine pairs with thymine, A to T, and cytosine pairs with guanine, C2G. So in those complementary strands, they're kept together by hydrogen bonds, and it was evident that this could contain the genetic material for life. However, it was unclear how these genes were expressed from the DNA. In other words, we could see that DNA held the material of genes and genetic material, but we didn't know how this was expressed, how it was translated to functional proteins, and basically RNA answered this question. And its discovery led to the discovery of the central dogma of molecular genetics, the flow of information through the cell. So let's see how that works. First, we have to start with a comparison of DNA and RNA. Now, there are similarities between DNA and RNA. They're both comprised of nucleotides. Now, a nucleotide is a phosphate, a sugar, and a nitrogenous base. The phosphate and the sugar makes up the backbone of the nucleotide chain. And the nitrogenous base is the adenine, thymine, guanine, and cytosine in the DNA. So these are the parts that will actually base pair with their complementary strand. However, there are important differences between DNA and RNA. While both DNA and RNA are made of nucleotides, these nucleotides differ in essential ways. First of all, sugar in RNA is ribose as compared to deoxyribose in DNA. Now, what this means is there's a certain carbon that instead of having an OH group or hydroxyl group, it has just a hydrogen. There's a loss of an oxygen in one of the carbons of deoxyribose as compared to ribose. Now, functionally, this makes DNA much more stable than RNA, which is one of the big reasons why it could not be the molecule that contains genetic material. The second difference is that RNA is generally single-stranded, whereas DNA is double-stranded. This goes back actually to the sugar. Deoxyribose in the DNA actually makes it much more able to base pair and create the double helix, which is very stable, as compared to RNA, which has the ribose, which makes it unable to, due to the structure itself, to base pair in such a way that it makes it a double strand, or at least very uncomfortable for the RNA to make a double strand. And the third important difference is that RNA contains uracil in place of thymine. That means that adenine will base pair with uracil instead of thymine when you're dealing with RNA. There are also functional differences between DNA and RNA. DNA contains all of the genetic material for a cell. You could relate it to a master plan of sorts in a building project. Say you have a building that you want to be built, and it is the master plan contained back at the architectural firm or the offices, whereas RNA molecules are small copies of specific portions of that DNA that code for polypeptides, which are also known as proteins. So RNA would be like the blueprint, or small, inexpensive copies of the master plan that you can take to the actual site to make the building. So in terms of the cell, DNA has all the genetic material to make proteins, and by extension, everything that an organism is. Whereas RNA molecules are small copies that can go out to the cytoplasm and ribosomes and make the proteins themselves. There are 3 essential types of RNA, and in this list I have the types and the functions of these RNAs. The first is messenger RNA, or mRNA. These are molecules that carry copies of instructions encoded in DNA to make protein. What this means is that the mRNA is the actual copy of the DNA portion coding for a gene. So you have the DNA, and the mRNA is a specific copy of that DNA which will code for a polypeptide. Ribosomal RNA, or rRNA, actually work to assemble those proteins. The actual organelle which assembles the protein is the ribosome. These are made up of several rRNA molecules and as many as 80 proteins. So the ribosome is basically this giant complex made up of rRNA, which catalyzes or initiates the translation of the mRNA strand into a functional protein. So it's what translates the code of the DNA. The third type of RNA is transfer RNA, or tRNA. These bring specific amino acids to the ribosome as outlined by the coded message in the mRNA strand. Now what this means is, on the mRNA strand there are codes, which I'll get into in another lesson, but there are specific codes on the mRNA strand. The ribosome brings in transfer RNA, which has an amino acid on it, and it complementary base pairs with that mRNA strand and attaches the polypeptides, but that comes later. But all you need to know right now is that tRNA brings specific amino acids to the ribosome as outlined by the coded message in the mRNA strand. Transcription. Now, transcription is the actual process by which mRNA is copied from DNA. Segments of DNA serve as the templates to produce complementary RNA molecules. Now there are differences between prokaryotes and eukaryotes. Prokaryotes, which are bacteria, transcription occurs in the cytoplasm because they have no membrane-bound organelles. They basically do not have a nucleus. They just have their DNA out in the middle of their cell. while eukaryotic transcription occurs in the nucleus. Now, as you should already know, eukaryotes do have a nucleus, do have membrane-bound organelles, and inside the nucleus, mRNA is transcribed from DNA, and in the next slide I'll go over how that is processed and how those differ, but transcription does occur in the nucleus for eukaryotes. The process by which this occurs is catalyzed by RNA polymerase, which is required to make RNA. RNA polymerase basically unwinds the DNA. It breaks the complementary strands, the base pairings, between the DNA strands, the two complementary ones that are wound in the double helix. It unwinds those and inserts itself into that DNA and uses one of the strands as a template for which it assembles nucleotides of the RNA. So essentially it unwinds the DNA at a specific portion when it reads or recognizes a certain promoter, and it understands that this is a portion of DNA which needs to be copied and translated and transcribed. Goes in there, copies that portion, and then detaches, and then there are other things that happen to that mRNA strand. Promoters, as I was talking about in the previous slide, RNA polymerase knows where to start transcription by attaching to promoters. A promoter is a region of DNA that has a specific sequence that is recognized by RNA polymerase. This means that the DNA itself has a certain sequence of bases that the RNA will recognize. Now, the RNA polymerase will go in, attach itself to the DNA at that promoter, and begin transcription from that point. So the promoter is necessary for transcription to even occur. There needs to be a signal to RNA polymerase that this is where transcription starts. There are also similar signals that tell transcription where to stop, but that is not, we're not going into that today. Another part is RNA editing. This is only important in eukaryotes. They have to undergo an editing process before they are allowed to leave the nucleus. One of these processing techniques are the splicing out of introns. Now, introns, which are non-coding portions or intervening sequences, are spliced out of the mRNA. Basically, there's a long mRNA strand, and these intervening sequences are cut out of the mRNA strand itself. The exons are then spliced together to form a complete mRNA strand. A cap is then added to the 5' end of the mRNA strand, and a tail is added to the 3' end of the mRNA strand. Only after mRNA goes through these editing processes is it allowed to leave the nucleus for eukaryotes. This means that RNA basically has to go through a maturing process before it can leave the nucleus and begin translation.